All right, next we're going to be looking at the humoral immune response. And the humoral immune response is when B cells encounter their target antigen, and that's what promotes this response from the B cells. And antibodies are produced from the effector B lymphocytes, which are plasma cells, and antibodies are going to be specific for that particular antigen that they were originally produced for. B cells are activated when antigens bind to their surface receptors. That cross-links the antigen and the B cell receptor and triggers the endocytosis of that receptor and antigen complex. So the antigen receptor complex will be endocytosed once they're cross-linked, and this is termed clonal selection. And that would lead to the proliferation and differentiation of B cells into effector cells. The effector cells of B cells are plasma cells. So the plasma cells are actually the antibody secreting cells. And they secrete specific antibodies at a rate of about 2,000 molecules per second for about 4 to 5 days, then die. And the antibodies that are created during that 4 to 5 days would circulate in the blood or lymph, binding to those free antigens that they recognize. And we would mark them for destruction by innate or other adaptive mechanisms. Here in figure 20.11, we can see that clonal selection. We have different B cells here, all with different B cell receptors, but this antigen only binds to the B cell receptor in this middle B cell. That would cause the activation of these B cells where they would proliferate to form a clone. After the cloning, we would get the activation to plasma cells. So that proliferation, then differentiation, is a part of that activation step. The plasma cells are the effector B cells, so we can see that they create antibodies, and these are the antibodies that would circulate in the blood or lymph. But we also see that the clones also produce memory B cells, which are primed to respond to the same antigen. So clone cells that do not become plasma cells become memory cells, and memory cells provide immunological memory, which is going to be able to mount an immediate response to future exposures to the same antigen. So here we see an example of a secondary exposure and a secondary response by the humoral immune system. So we have here a memory cell, a clone of cells identical to ancestral cells, that is challenged by a subsequent exposure to the same antigen. But this would result in a larger, more rapid response. We would get more plasma cells, more antibodies secreted, and more memory cells. So we can compare a primary immune response to a secondary immune response. A primary immune response is wherein cell proliferation and differentiation upon exposure to an antigen for the first time has a lag period of 3 to 6 days. And peak levels of plasma antibody production are going to be reached in about 10 days, and then antibody levels will decline. With a secondary immune response, this is a re-exposure to the same antigen, but we get a faster and more prolonged, even more effective response because we have sensitized memory cells that are providing us with immunological memory. So we get a response within hours, not days. We get antibody levels that are way higher than the initial primary response levels that peak in 2 to 3 days, not in a couple of days, up to 6 or 10. Then the antibodies are going to be able to even bind with greater affinity. Greater affinity means they bind better to the B cell receptor, and that's going to lead to activation and proliferation differentiation that allows antibody levels to remain high for weeks to months. So let's look at figure 20.12, comparing primary and secondary humoral responses. We see that the primary immune response to antigen A occurs only after a delay, whereas the secondary immune response to antigen A is much faster, much larger, and that the primary immune response to a similar antigen but slightly different antigen B is similar to that of antigen A. So it's not a matter of when an individual got infected by an antigen and the type of response that they get. It's whether or not this individual has seen this antigen before, whether or not their body has immunological memory because of the generation of memory cells during that primary immune response. Next, we're going to look at active and passive humoral immunity. So we're still focusing on B lymphocytes. Active humoral immunity occurs when B cells encounter antigens and produce specific antibodies against them. So essentially, active humoral immunity occurs when B cells are challenged, when they're activated, when proliferation and differentiation occurs. There's 2 types of active humoral immunity, naturally acquired and artificially acquired. Naturally acquired active humoral immunity is formed in response to an actual bacterial or viral infection. So when B cells are activated and then they proliferate and differentiate because of a bacterial infection, that is an example of naturally acquired active humoral immunity because the B cells were actively challenged. We had to turn the B cells into plasma cells in order to get antibodies. But there's also artificially acquired active humoral immunity. And artificially acquired active humoral immunity is similar to the naturally acquired, but we're going to only have a response, meaning antibody production, because of the introduction of a vaccine or dead or attenuated pathogen into a human body. So we kind of skipped the actual bacterial or viral infection and just introduced something that is immunogenic and reactive. And this would provide the antigenic determinants that are going to cause that immunogenic and reactive response, but it spares us the symptoms of the primary response. So because of the artificial nature of vaccines, and I'm saying artificial in that they are created by humans, so they're not created by the human body, but they do use certain molecules in the human body, which is essentially just the dead bacteria or viral infection. Because of that kind of artificial nature, we're going to call vaccines an example of artificially acquired active humoral immunity. Whereas naturally acquired active humoral immunity is just a general bacterial or viral infection that stresses B cells to become plasma cells and produce antibodies. So keep in mind that for artificially acquired active humoral immunity, that the vaccines or whatever is being injected is kind of lab acquired. For passive humoral immunity, there's also naturally and artificially acquired, but passive humoral immunity is different than active humoral immunity. With passive humoral immunity, we're going to see an occurrence in which ready-made antibodies are introduced into the body. So we're not challenging B cells. We're not making B cells become activated, proliferate, differentiate, and produce antibodies. Wherein we were seeing that with active humoral immunity. For passive humoral immunity, we're skipping the B cell part and we're just introducing into the human body already made antibodies. So go ahead and kind of link in your mind passive humoral immunity with ready-made antibodies. So B cells are not challenged in this example. Immunological memory does not occur. They would with active. So there's two different types, and protection would end when the antibodies that are ready-made and introduced to the body degrade. But the naturally acquired type of passive humoral immunity is when antibodies are delivered, say, to the fetus via the placenta or to an infant through milk. The artificially acquired would be an injection of serum, such as gamma globulin. A great example of artificially acquired passive humoral immunity is the use of antivenom. Even though the body could fight off the effects of snake venom, for example, snake venom works so quickly that the body does not have time to prime its adaptive response. So we artificially inject a serum that contains ready-made antibodies against the snake venom, and that would provide immediate protection, but that protection would end when those ready-made antibodies naturally degrade in the body. But by that time, the adaptive defense system of the individual who was bitten, say, by a snake would have started to produce their own gamma globulin against the venom that was injected by the snake. So again, another example for artificially acquired passive humoral immunity is antivenom. And then we looked at two examples there for naturally acquired passive humoral immunity, and that was antibody delivery through the placenta to a fetus or to an infant through breastfeeding. So let's just look at all of these one more time, because when you say the terms, they get long. For example, active humoral immunity, passive humoral immunity. Now let's add the other terms. There is naturally acquired active humoral immunity. There is artificially acquired active humoral immunity. There's also naturally acquired passive humoral immunity and artificially acquired passive humoral immunity. So those terms get long, but we can kind of break down the differences in each. So naturally acquired means that there is a natural source either to the activation of B cells or to the introduction of ready-made antibodies. For artificially acquired, there is a lab component or an external component that is ready to introduce a challenge to B cells, such as with a vaccine, or a serum of ready-made antibodies, such as with antivenom. Examples of naturally acquired active humoral immunity would be someone getting an infection, just kind of a natural infection with a bacterium, for example. An example of artificially acquired active humoral immunity would be a vaccine, which contains dead or attenuated pathogens, which introduce the antigen, especially the antigenic determinants of a bacterium, for example, to the body, but don't necessarily introduce the symptoms. An example of naturally acquired passive humoral immunity would be passing antibodies through the placenta to a fetus or to an infant via breastfeeding. And then an example of artificially acquired passive humoral immunity would be the use of antivenom, which is an injection of exogenous antibodies. Let's talk more now about antibodies. They're also called immunoglobulin, or IGs for short. Immunoglobulins are of different types. They're capable of binding specifically with an antigen detected by a B cell during clonal selection. And there's actually 5 types, or 5 IG or immunoglobulin classes. Also, don't forget that antibodies are proteins, and these proteins are being secreted by plasma cells, and plasma cells are effector B lymphocytes. The basic antibody structure is a T or Y shape. They tend to be monomers, or we tend to see when we draw one out just one antibody, but they can form complexes. We can have 5 or 2 antibodies come together, and we'll look at those examples. But overall, if we're looking at just one antibody monomer, it's going to have a T or Y shape. It consists of 4 looping polypeptide chains, and they're linked by disulfide bonds. The 4 chains contain 2 heavy and 2 light chains. The heavy chains are longer than the light chains, and on one side we're going to have variable regions, and on the other side there's going to be a constant region. The variable regions are where we would find antigen binding sites, and the antigen binding sites are going to be not only located in these variable regions, but if we look back at that Y shape, they're located at the 2 top pieces of the Y, or the superior arms of the Y. The stems are the constant region, and the constant region is going to determine the antibody class, and they're considered the effector region of the antibody because it serves common functions in all antibodies, dictating not only the types of cells or chemicals that an antibody can bind to, but how antibody classes function in order to eliminate antigens. An example would be some antibodies are able to fix complement, while some can't, and some circulate in the blood, whereas others are found in body secretions, and that is determined by this constant C or stem region. So let's look at an antibody monomer. This is figure 20.14a in your lecture textbook. First, let me point out the heavy chain. So we have one heavy chain here in the middle, another heavy chain here, and then we have 2 in purple light chains. And we have 1, 2, 3, 4 polypeptides. So these are just specifically arranged small proteins. The specific arrangements are linked by these yellow disulfide bonds. There's actually an area in the center with double disulfide bonds, and that is the hinge region. We also are noting 1 antigen binding site, but be aware that there's another antigen binding site over here. So 1 antibody monomer contains 2 locations for an antigen to bind to it. So if I said I have 5 antibodies, how many antigens can I bind? You would have to take how many antibodies you have and multiply that by 2. So I would have 10 locations for an antigen to bind if I had 10 antibodies. So we'll do another example of that coming up. The antigen binding sites are located on the tips of this Y-shaped monomer. They are in the variable region, so these kind of dark stained regions versus the light stained regions. And then down here below the hinge region is the stem or constant region. The different antibody classes include IgM, IgA, IgD, IgG, and IgE. And they spell a name, which is MAG. So if you need a way to remember the 5 major classes of antibodies, you can just remember the name MAG, M-A-D-G-E. We're going to look at each one. So IgM can form a pentamer. So that's going to be a complex of 5 antibodies. It's much larger than other complexes that we'll see. And IgM is also the first antibody released. And because it can form a pentamer, this complex becomes a potent agglutinating agent. When we see a picture of it, I'll show you how. But agglutination is... So this is going to be a potent agglutinating agent. It would work in mismatched blood transfusions. It's readily able to fix and activate complement. Then you have IgA, which is also called secretory IgA. It can be a monomer, but it can also form a dimer. It's a little bit smaller than the pentamer that IgM can form, but we find secretory IgA in mucus and other secretions. That's why it's called secretory IgA. It is able to help prevent the entry of pathogens into deeper tissues. So here we see 2 immunoglobulin classes, IgM and IgA, both of which can form complexes. A complex is just multiple monomers of an antibody coming together. So here we have our pentamer. And if this was floating around in the blood and it came into contact with antigens that it recognized, it could bind 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Because I have 5 antibodies and each one has 2 locations to which it can bind an antigen, if I have an IgM pentamer, I can bind to 10 antigens. So I said we were going to go over that again, and that's what we're doing right now. Again, this is the first immunoglobulin class secreted by plasma cells, essentially during the primary response. So it is diagnostically useful because the presence of IgM in the plasma usually indicates a current infection by the pathogen that's eliciting essentially the primary immunoglobulin response. We know it readily activates complement, but other than that, it's really the fact that it can form a pentamer that is most important because that makes it a great agglutinating agent because it could bind to 10 antigens at once. It would hold 10 things in one location. It would clump them all together. IgA is also able to exist as a monomer, but what we see here is a dimer. And if we count how many antigens this dimer could hold, we have to count each antigen binding site. So 1, 2, 3, 4. This dimer could bind to 4 antigens because each of these 2 antibodies contain 2 antigen binding sites. It's also called secretory IgA, and it tends to be found in mucous membranes and secretions. The other classes are IgD, IgG, and IgE. IgD is generally a monomer. We know that it functions as a B cell receptor. IgG is going to be secreted or made during secondary or late primary responses. So during a primary response, we can see a change in that constant region from IgM to IgG. But IgM is always produced first, but we may see changes to that as a primary response continues. We also know that IgG can cross the placental barrier. And then IgE, which is generally a monomer, is active in some allergic and parasitic infections. A way to remember that is IgE. The E is similar to eosinophils, which are also active in some allergic and parasitic infections. And they generally cause mast cells and basophils to release histamine. This is what we're seeing here is monomers of IgD, G, and E. We see D, which is a B cell receptor, G, which is produced during a secondary response or late primary response, and E, which is going to help mediate inflammation and allergic reaction. Now, I said this before, but plasma cells can switch from making one class of antibodies to another. We can change that constant region during a primary, even a secondary response, even though it's common in a primary response. We still retain the specificity for the same antigen because we're not changing the antigen binding sites, we're just changing the constant region. So for IgM, which is released during a primary response, we may see the amount of that wane during a secondary response or in a late primary response because IgM can be switched to IgG. And almost all secondary responses would release IgG initially because in a secondary response, we would release IgG as opposed to IgM. And switching from IgM to IgA or IgE can also occur. Next, we're going to look at what do antibodies target? And when they target something, how do they function? And antibodies are not going to destroy antigens, they're just going to bind to them. But when they bind to them, they can inactivate them, they can tag them, they can form what's called an antigen-antibody complex, which is just the antibody binding to its antigen. But depending on the type of antibody we're looking at, we can get defensive mechanisms that equate to things like neutralization, agglutination, which is clumping, precipitation, which is turning something that was soluble to insoluble, and even complement fixation. So we're going to look at each one of those. Probably the simplest to understand is neutralization, because we've talked about opsonization. And neutralization is one of the most important defensive mechanisms that antibodies are capable of. Antibodies are able to block specific sites on viruses or even bacterial exotoxins by opsonizing them, by coating them, by preventing antigens from these particular viruses or bacterial exotoxins from binding to receptors on tissue cells. And so forming an antigen-antibody complex or opsonizing a particular virus or bacterium is going to neutralize, in this instance, that particular antigenic determinant. Agglutination is when antibodies can bind the same determinant on two different antigens at the same time. So IgM is great at this because it has 5 monomers bound together. And so we can definitely bind multiple antigens on the same determinant. And this allows for antigen-antibody complexes that are cross-linked. We get lattice-like clumps, so we get agglutination. That's what agglutination is, the clumping of antigens. This can occur during a mismatched blood transfusion. Precipitation is where we take something that was dissolved in a fluid, such as, for example, certain plasma proteins, and we make it insoluble. We make it more solid or visible to the immune system. So precipitation is when soluble molecules, instead of cells, are cross-linked into complexes with antibodies. And these complexes are easier to see, easier to recognize by phagocytose-capable cells, and they're able to basically be not only precipitated out of solution, but then attacked by phagocytes and engulfed. And then there's complement fixation and activation. And this is a main antibody defense against cellular antigens, such as an entire microbe or bacterium, or mismatched red blood cells. And so when several antibodies are bound close together on the same antigen, that kind of lines up a certain array of proteins that are perfect for complement. For complement to bind to these sites, specifically on the stem regions of antibodies, that alignment is required. And the alignment triggers complement fixation, and that would lead to cell lysis. It would lead to the amplification of inflammatory responses, and it could lead to opsonization. All things we've talked about before when we looked at inflammation and complement fixation. I highly recommend looking at figure 20.15, which goes over the mechanisms of antibody action. They're going to start with an antibody binding to its antigen and forming an antigen-antibody complex. Now, these antigens must be specific for these antibodies. The complex is able to neutralize, agglutinate, or precipitate substances, whether it be a whole microbe or part of a microbe, and that will enhance phagocytosis or engulfing by phagocytosis, inflammation, which includes the release of histamine, which causes vasodilation and increased capillary permeability, as well as phagocyte mobilization. So that's why we have kind of an arrow going back here. And then we also see that it can trigger complement activation if the antibodies line up a certain way. Complement activation will lead to the generation of C3, which can be cleaved to form C3A and C3b. C3b can act as an opsonizer. It can cause cell lysis, and C3A can enhance inflammation. So that's what we're seeing here is the use of C3b as well as the membrane attack complex to cause cell lysis. So this is an overview of those mechanisms of antibody action that we just got done looking at. And clinical or homeostatic imbalance we also want to mention is parasitic infections from worms, such as there's Ascaris and Schistosoma, and they require a different immune attack strategy. So worms are too big for kind of regular antibody mechanism attacks, which could be abbreviated PLAN for precipitation, lysis, which is by complement, agglutination, or neutralization. For these parasitic infections, we would need something different. We would need something like IgE antibodies to play a critical role in the destruction of worms by binding to the surface of the worm and then marking it for destruction by eosinophils. We know eosinophils have a role in allergy and parasitic infections. Eosinophils are actually able to bind to the exposed stems of IgE, and that triggers eosinophils to release their granules. Their granules contain toxic contents, and that goes onto the prey, in this case, the parasite, and that would lyse it from the outside. Something else we want to mention is research into monoclonal antibodies, specifically as clinical and research tools. Now, monoclonal antibodies are just commercially produced antibodies, but they're pure. They're made of one particular antibody that is specific for a single antigenic determinant. They're produced by hybridomas, which is a cell hybrid formed from a fusion of a tumor cell and a B cell. So essentially they take a cancer-like cell that can only produce one type of cell, and they fuse it with a B cell, so we only get this one type of B cell, which can turn into a plasma cell that produces one type of antibody. So the tumor cell portion allows cells to proliferate indefinitely, while the B cell portion allows production of a single type of antibody. And these monoclonal antibodies are used in research, clinical testing, and cancer treatment for the recognition of particular antigens or location of particular antigens. And then if we were to summarize our antibody actions, we would say that forming an antigen-antibody complex is not meant to destroy an antigen. Instead, we would use them to prepare the antigen, whether it's on a microbe or it's just complexing together with that antibody, prepare those things for destruction by innate defenses. The types of things we talked about were neutralization, precipitation, lysis by complement, and agglutination. And antibodies tend to go after the extracellular pathogens, so they're not going to invade solid tissues. They're not going to go into cells. And unless there's special circumstances where a lesion or opening is present to a cell, there is a recent exception there found. We do know that antibodies can act intracellularly if they're attached to a virus and they're attached to that virus before that virus goes into a cell. Then they can actually work inside that cell, but it's a rare occurrence and it's a recent exception that's found. And that's it for part B. Make sure to look at part C as well, where we'll look at the cell-mediated immune system of the adaptive defense system in more detail.